Hemophilia is a genetic disorder which disrupts the body's process of blood clotting. This can lead to excessive or spontaneous bleeding. Due to the prevalence of hemophilia A, or factor VIII hemophilia, in both prevalent families, this is the specific type of hemophilia we will be focusing on in this particular case. This differs from hemophilia B in a specific protein deficiency which it is caused by. Hemophilia A is caused by mutations in the F8 gene, located on the X chromosome. In absence of any abnormality, the gene provides instructions for the production of a protein called coagulation factor VIII. The mutated gene, however, produces either ineffectual proteins or simply none at all. Depending on the severity of the patient's case, these proteins are then limited in their ability to participate in the blood clotting process, or are unable to at all. This is also what determines the severity of the various symptoms of hemophilia, which will be covered later. Hemophilia is an X-linked recessive trait. This has a number of implications regarding the way in which the disorder is passed down. As it is an X-linked trait, fathers cannot pass it on to their prospective sons, as it does not appear on the Y chromosome contributed in this scenario. In addition, this means males are much more likely to receive the disorder, as they have only one X chromosome, meaning they only need to receive one carrier allele to have hemophilia. This is in contrast to females, who must receive two alleles to receive the disorder. Finally, the recessive aspect of hemophilia makes it so that an individual must receive only the hemophilia allele, either one for males or two for females. Hemophilia can be recognized by a number of symptoms, many of which involve excessive or continuous bleeding. For example, bleeding of the mouth and gums after tooth removal, frequent and subsiding nosebleeds, as well as tightness and swelling in the joints caused by internal bleeding. Internal bleeding is often the most severe consequence of hemophilia, as it can damage organs and in serious cases prove life-threatening. Symptoms such as double vision, prolonged headaches, and seizures can be a sign of a brain bleed, a rare but extremely serious product of hemophilia. All these symptoms are caused by the absence or limited number of effective coagulation factor VIII proteins. This is the family pedigree for Greg and Olga, assuming neither of them themselves have hemophilia, as would almost certainly be disclosed during a consultation. As Greg is a male and unaffected, we know his genotype. As for Olga, we know her father is unaffected. Olga's maternal grandmother has to be a carrier, as we know her husband was unaffected and they produced Olga's maternal uncle, who has hemophilia. This is the only way that this is possible. In addition, we know Olga's mom is a carrier as well, because she produced an affected son with an unaffected husband. It is impossible for Olga to be affected, because of the assumption discussed before, and that this would require both parents to be hemophiliacs. Therefore, Olga is unaffected, but there's a 50-50 chance she is a carrier. In the first scenario, Olga is an unaffected homozygote. This will result in her children being 50% unaffected male and 50% unaffected female, none carrying a hemophiliac gene. In the second case, Olga is an unaffected heterozygote. This would result in her children being 25% unaffected male, 25% unaffected female, 25% unaffected male, and 25% unaffected carrier female. There are two definitive prenatal tests available to determine the possible presence of hemophilia in an unborn child. The first is chorionic villus sampling, or CVS, which involves inserting a needle into the vagina and extracting DNA from the placenta, which contains identical DNA to that of the fetus. This can then be used to determine whether or not the child will have hemophilia, and is the most popular method of prenatal diagnosis. Alternatively, amniocentesis can be performed, which involves insertion of a needle into the abdomen and the retrieval of amniotic fluid, which contains cells from the fetus that can then be analyzed. Once a child is born with hemophilia, multiple treatment options are available, the most common of which is called replacement therapy. This involves injecting blood with highly concentrated amounts of the missing protein, F8, in the case of hemophilia A. In severe cases, the treatment often must occur regularly, called prophylaxis, whereas in mild cases the treatment occurs only when needed. This option, although very effective, has downsides. Antibodies can develop which prevent the treatment from working properly, and it can sometimes be delayed and thus ineffective if not performed on a regular schedule. Replacement therapy is also a more significant time commitment and could be an inconvenience. Another option is desmopressin, a synthetic hormone which increases the production of proteins and allows F8 to stay in the blood longer. It is used exclusively in cases of mild hemophilia A and often administered through injection. This is less invasive and costly than replacement therapy, but its effects are short-lived and thus its uses are somewhat limited. Another option is amniocaprotic acid, which can be used to prevent the breakdown of blood clots and is taken through a liquid or pill form. This is often used in conjunction with demand therapy for mild hemophiliacs to first form and then maintain a clot. This option is the most convenient and relatively inexpensive, but its effectiveness on its own is less widespread than previous methods, as it only maintains clots that are already formed. The treatment options I would recommend for the child of Greg and Olga would depend on the severity of their condition. If mild, desmopressin along with amniocaprotic acid would be sufficient. For moderate hemophilia, demand replacement therapy along with desmopressin is necessary. Finally, if severe, scheduled replacement therapy multiple times per week would be necessary.